Welcome to the Atlantico podcast, where we talk about the science behind the Atlantico project, the Atlantic Ocean, and the human adventures experienced along the way. Here, we have conversations with guests from around the world who work together so that we can better understand, manage, and protect the ocean. So let's embark on the journey of Atlantico and discover the world that lies above and beneath the surface of the beautiful Atlantic Ocean. Welcome back to the podcast. Today we discover a region of the Atlantic with Flora Vincent. Flora was the chief scientist on board of Tara during one of the sampling studies of Mission Microbiomes, and she will tell us about her experience and explain what scientific questions the team has been tackling while at sea. Flora, welcome to the podcast. We are very happy to have you on the show today.
Hi, Louise. Hi, everyone. And thanks for hosting me.
It's a pleasure. So, to begin each episode, we like to get to know our guests, to understand how their connection with the ocean started and what their journey has been to where they are today. So can you tell us about you, about your story and what you do today, and a little bit about your journey?
Yeah, sure. So I was born in south of France, next to the sea. And actually at 8 years old, I learned how to scuba dive. And I really think that this is when I got hooked. Because I could literally spend hours under the water looking at, all the different kind of fishes, studying octopus behavior and really trying to find the right algae. And for many years, I was actually a scuba diver instructor during the summer. And this is really probably when my curiosity for biology and life science really started. So after that, I was trained as a life science engineer. And I actually decided to become a researcher after spending a gap year working in marine biology in Japan and in French Polynesia. And it's actually during this gap year that I discovered Tara back in 2011. And for me, it was really a revelation, mind-blowing to know that this project existed. So I did a PhD in France, working on the Tara expedition, followed by a postdoc in Israel, where in the end I also reconnected with the Tara boat again. And in general, I'm interested in really the community of tiny beings, and more particularly those marine microbes that we call protists. These marine microbes are really invisible to the naked eye, but they really represent an impressive diversity and richness of organisms. So those protists are neither bacteria nor viruses. They actually form a third large family of really underestimated marine microbes that are actually super important for our ecosystem. They produce a lot of oxygen. They are an important source of food for other organisms. And I'm particularly interested in the social life of those microbes. For instance, how do these various microbes interact with each other? How do these interactions affect the development of all those microorganisms? And of course, how will the environment and future climate change actually impact those interactions? And now I'm opening my own lab at the EMBL in Germany to continue spying on the social life of microbes.
That's amazing, and congratulations on opening your own lab. That's an amazing achievement. So today we're here to discover about your experience during one of the scientific studies of Mission Microbiomes. You've talked to us about, your connection with Tara. That is one of them. You were on board of Tara from Buenos Aires to Ushuaia along the coast of Argentina. And you had a collaboration with the Argentinian schooner Husey as well during that time. So maybe to start with, you could tell us the scientific questions that you were addressing during that leg, why it was so important to study what you were studying, and how it will contribute to increasing our knowledge of the Atlantic Ocean.
So every year in Argentina, there is a really impressive event that happens in the ocean where, under good conditions of light and nutrient, small microalgae start to grow very, very fast. And they proliferate in mass to form what we call a bloom. And this bloom is so big that you can literally see it from space because it kind of changes the color of the ocean. And part of those microalgae are called coccolithophores, which is a very cute name. But those coccolithophores have a hard life when they bloom because they can be killed by viruses, by bacteria. They can be eaten by small crustaceans. And some of those coccolithophores will survive. Some will sink. For example, when you see the White Cliffs of Dovers, all of these are basically dead coccolithophores. And our goal was really to follow the initiation and termination of this bloom, to understand how all those interactions between coccolithophores and other microbes will impact the ecosystem. Because the truth is that we still today don't really know why a bloom ends. And we implemented really state-of-the-art methods to try and better understand how blooms are terminated. And for example, in the future, this could actually help to also better monitor the health of our planet. So yeah, as you mentioned, the mission was done in collaboration with Argentinian scientists and coast guards. And we were very lucky because a month before Tara was in Argentina, the Husey, which is the Argentinian scientific vessel, sampled the same area, but in the other direction. So from Ushuaia to Buenos Aires. And the big added value of this is that from now we will know the conditions that were there before the bloom started. And that's actually super important to understand why the bloom started in the first place. And most importantly, on the Tara side, on the Tara side, we had the expertise of studying coccolithophores in the lab, let's say with fancy tools and approaches. But on the Argentinian side, they really had the expertise of the area, which was actually super critical because even if you see those blooms from space, finding them in the ocean is a bit like looking for a needle in a haystack. And this is why we called it a coccolithophore chase or a hunt, because every day there was a strategic decision and a whole process on where we would go next. And conducting chases like this in real time is extremely challenging, but it's also super, super fun.
Nice. It sounds very exciting. So how did you do it? How did you study those coccolithophores? What did you have to do? And also what will happen next with everything that you've collected on board?
So to study those blooms, you actually first need to find them. And to do that, we used very different methods that cover very different spatial scales. For example, we started with satellite data that can help you to define a rough area of where to go. And then once the boat is in the area, you can use, or we use machines on board to specifically quantify the presence of those coccolithophores. And then we use different sensors to see at which depth we need to collect the water. So there's a lot of things that you learn on the way. And once we found let's say the perfect location and the perfect depth at which to sample, we had to collect hundreds of liters of water, 200 to 300 in just one morning. And then it's really a kind of choreography that starts on the boat because everyone knows what he or she has to do. Sometimes you need to filter water. Sometimes you need to add some specific chemicals. Some of it gets stored in the fridge, in the freezer. Some stays at room temperature. So there's really all those different job posts that starts. But this is only the collection phase because what happens next is that those samples will get shipped in different labs that collaborated on this mission. And then we'll start the whole work of processing the samples and then analyzing the data. So in a sense, even if the expedition ended 6 months ago, the real job will only start in the coming weeks.
Yeah, and I guess it will take some more months after that to get to even first results and to answer those big questions that we ask ourselves. And each of the legs of the expedition is dedicated to a woman involved in marine sciences. And in this case, it was dedicated to Anamaria Gashiosso. Can you tell us a little bit about her and her contribution to the field of marine sciences?
Anamaria Gashiosso was an Argentinian scientist, a marine biologist, and she actually played a really important role in characterizing phytoplankton species in this whole area of Argentina between Buenos Aires and Ushuaia. So it was really important for us to really help and make her name visible and known, to also recognize her contribution to science in that area. And this is something amazing, I think, with the Atlantico mission, that every small topical study is named after a women scientist that contributed to the local knowledge. And in her case, Anamaria Gashiosso was actually the first one to identify that there was a bloom of coccolithophores. So we could see those coccolithophores from space, but we didn't really know what they were. And she's the one that actually managed to identify that these were coccolithophores. And this, again, is a really amazing contribution from the local Argentinian scientists. And they came up with that name. And then, I said, there is no Wikipedia page. We need to create a Wikipedia page. And then I collaborated with Valeria in Argentina, who actually really helped me to fill up this page with actual content. So yes, it's important for us that this is named after Anamaria Gashiosso, and we're actually pretty happy because we think that making her name visible, I think it worked.
Excellent. Yeah, it's one of our kind of side focus here in Atlantico to try and make women scientists more visible. Those that work on the project, to give them opportunities, but also, people in the past that have helped to advance the field of marine sciences. So that is just one of the ways. And finally, maybe more about your personal experience on board Tara. Can you share what it was like for you, about your life on board, and the memories you've brought back from that region of the Atlantic?
It was the third time for me on the Tara schooner, but the first time as a chief scientist. So the experience is actually very, very different from the previous ones. Of course, the experience of life on board is always fantastic, and it's always a lot of fun. We had an amazing crew of sailors and scientists, super committed into making this hunt work out. We would play games every day on how many coccolithophores were in the water, and everyone was like super motivated to win. But also when it came to the science, their input, the input of sailors, was really instrumental. So we would sit every day and discuss satellite pictures together and think about what was the most efficient, but also sustainable way to get there. And in general, the sailors really pushed us to better practices, for instance, by recovering some drifters that we release in the water to track the bloom. So they really care about the health of the ocean, and they really push us scientists to do better science. I also thought quite a lot about what type of chief scientist I want to be, especially because it was the first time. And the very useful advice that someone gave me was the following. This person told me, your goal is that no one needs you by the end of the expedition. So at the beginning, there was a lot of background work on setting up, when do we meet and what is our decision process? How do we provide feedback and share information? Who does what? So basically, I was putting a lot of oil in the different parts of the machine, mostly dealing with emergency and complaints and frustrations. But a major achievement is that by the end, all of us felt like decisions were really taken in a collective manner, and that every voice was heard. And that for me was super, super important. I also think that I was very lucky because the team of scientists on board was really excellent. I knew all of them from before, all very driven, competent, working beasts. I had to tell them to rest. So in a way, I'm super grateful that this is, at least this first experience to lead a team on board was done in this context. And I think that if there's a major memory that I keep, from this region of the Atlantic, it's definitely sailing on the Beagle Channel on a scientific boat at the end of our trip. Because I couldn't stop thinking that Charles Darwin was there in 1833, and that when Charles Darwin went through the Beagle Channel, this trip probably transformed the way we think about biology and how species evolve. So I really felt like I was also standing on shoulders of giants.
Wow. Amazing image, and I guess you could really feel that history in the place you were in. Amazing. Flora, thank you so much for sharing all of this today with us. It was very interesting. As always, I keep learning every episode, and I hope that everybody listening is always is doing the same, learning and getting inspired to learn more about the ocean, but also to get involved and engage to protect the ocean. So in the show notes today, we will put the link to the video which was made during your leg of the expedition. And also we produced an article, well, you wrote an article that is published on our website. So there's a couple of images there you can have a look at. And of course to the Wikipedia page that you put together with some help from the Argentinian team. We'll put the link to all of that in the show notes. And I think once again, just thank you very much, Flora.
Thank you very much.
We hope that you've enjoyed today's episode and look forward to seeing you next time. You can follow the Atlantico project on our website on www.atlantico.eu, and you can also find us on Twitter, Instagram and Facebook. All the links and information on the project and on today's episode is in the show notes. Atlantico is a project funded under Horizon 2020, a European Union research and innovation programme.